Macrophage phenotype. Macrophages have long been considered to be important immune effector cells, which are first identified by Elie Metchnikoff as phagocytic cells. Macrophages help to eliminate pathogens, and are also involved in the removal of cellular debris that is generated during tissue remodeling, and rapidly and efficiently clear cells that have undergone apoptosis. This clearance process is a vital metabolic contribution that occurs independently of immune cell signaling, and the removal of effete or apoptotic cells seems to result in little or no production of immune mediators by unstimulated macrophages. Necrosis that results from trauma or stress also generates cellular debris that must be cleared by macrophages. The clearance of this debris leads to the markedly dramatic changes in their physiology, including alterations in the expression of surface proteins and the production of cytokines and inflammatory mediators. The alterations in macrophage surface protein expression in response to these stimuli could potentially be used to identify biochemical markers that are unique to these altered cells. Macrophages have remarkable plasticity that allows them to efficiently respond to environmental signals and change their phenotype. Their physiology can be markedly altered by both innate and adaptive immune responses. The term macrophage activation, which refers to classical activation, was introduced in the 1960s in an infection context to describe the antigen-dependent, but non-specific enhanced, microbicidal activity of macrophages toward Bacillus calmette-guerin and Listeria, upon secondary exposure to the pathogens. In 1973, researchers found that independent cellular factors could also promote resistance of infection without involvement of pathogens. Almost at the same time, lymphocytes were identified as the major antigen-specific cells responsible for microbicidal activation of macrophage. In 1989, when the heterogeneity in the helper T cell compartment was subsequently reported, Mosvernen and Kaufman reviewed the different functions and lymphokin secretion between two types of cloned helper T cells, and proposed the concept of Th1 and Th2. With the discovery of upregulation of macrophage mannose receptor, MRC1, as a specific marker of interleukin-4/interleukin-13 activated macrophages, which was coupled with the enhanced expression of MHC2, the concept of alternatively activated macrophages, also known as M2, was first proposed. The M1 stimuli are grouped according to their ability to induce prototypic inflammatory responses and markers, but their source, role, receptors, and signaling pathways differ substantially. If gamma is the main cytokine associated with M1 activation and the main Th1 cell product. If gamma binds to its receptor, which recruits JAK1 and JAK2 adapters that activate STAT1 and interferon regulatory factors, pathogens are recognized by pattern recognition receptors. LPS is the best studied M1 macrophage signal and is recognized by Toll-like receptor 4. The profile lights polypeptide gene enhancer nuclear factor kappa, activator protein 1 and interferon regulatory factors. Produced by a variety of cells, granulocyte macrophage colony stimulating factor recruits JAK2, leading to the activation of STAT5, ERK, and AKT, as well as the nuclear translocation of NF-kappa B and IRF5 upon binding. The M2 group of stimuli arose from the initial interleukin-4 observations, and they are grouped mainly due to their ability to antagonize prototypic inflammatory responses and markers. Interleukin-4 is produced by the Th2 cells, eosinophiles, basophiles, or macrophages themselves. Receptor binding of interleukin-4 activates JAK1 and JAK3, which further leads to STAT6 activation and translocation. Glucocorticoid hormones are metabolized by cellular enzymes in macrophages, and then diffuse through the membrane to bind the glucocorticoid receptor alpha, leading to nuclear translocation of the complex. The complex binds DNA directly to promote or repress gene transcription, or indirectly by interacting with transcription factors, such as NF-kappa B or activator protein 1. Interleukin-10 binds the interleukin-10 receptor. Receptor autophosphorylation leads to the activation of the transcription factor STAT3 and its binding mediates inhibition of pro-inflammatory cytokine expression. The definition of M1 and M2 macrophage polarity derives from the pre-genomic era, when a few markers were considered to establish differences and similarities in macrophage responses to stimuli. M1 phenotype macrophages express numerous pro-inflammatory mediators, including TNF-alpha, interleukin-1 and 6, reactive nitrogen and oxygen intermediates. M1 macrophages play an important role in Th1 responses, type 1 inflammation, killing of intercellular pathogens and tumor resistance. M2 macrophages express molecules including resistin-like alpha, also known as Fis1, arginase 1, chitinase 3 like 3, also known as YM1, interleukin-10 and MRC1, also known as CD206. They are supposed to be involved in Th2 responses, type 2 inflammation, allergy, parasite infestation, tissue remodeling and tumor progression. For more information, please visit creativediagnostics.com.